Culturing of fathead minnows. The fathead minnow is used in toxicity tests at various life stages. Embryos, larvae, and juveniles are the life stages used most often. Familiarity with the specific needs of every life stage is needed to produce healthy cultures. Fathead minnows adapt readily to a wide variety of culture conditions. Therefore, there isn't one set of conditions that's exactly right for every laboratory. This video presents methods developed over the last 20 years at the EPA's Mid-Continent Ecology Division in Duluth, Minnesota. Other successful methods exist, and modifications to the methods may be required for specific circumstances. Because healthy animals are critical for successful toxicity testing, laboratory personnel must be familiar with proper handling and culturing procedures. This video presents recommended conditions and methods for the following parameters: choice of culture water, initiating cultures, breeding and feeding the fish, collecting embryos, and producing animals for test use. First, choose a type of culture water and make arrangements for the culture. One of the most important considerations is the water source. Two options are to use either a natural or a synthetic water, and the objectives of the test should be considered before deciding which to use. Regardless of the selection, the culture water should be reliable from a consistent source and similar to the water being used for testing. Natural waters are generally a surface water, well water, or spring water. Before use, check the water for pesticides, heavy metals, sulfides, fish disease vectors, and any other suspected contaminants. Techniques for measuring the parameters are described in the American Public Health Association's Standard Methods Handbook. Natural water is recommended, but reconstituted water or dechlorinated tap water can be used. The EPA manual, Short-Term Methods for Estimating the Chronic Toxicity of Effluents and Receiving Waters to Freshwater Organisms, referred to as the Chronic Manual, describes the reagent-grade chemical concentrations necessary to obtain the desired reconstituted water type and hardness. The diluent water for the reconstituted water should be filtered through a high-grade filtering system with the cartridges arranged in the following order: ion exchange, carbon, organics Q, and a fine filter. Once prepared, all waters must be aerated for 24 to 48 hours until the pH meets the minimum specifications as described in the Chronic Manual. Laboratories are required to perform a reference toxicant test each month on the water used for testing. The type of reference toxicant test to use is dependent on the intended use of the cultured fish. For instance, if subchronic tests are the primary use of the organisms, a subchronic test must be run with a standard reference toxicant. Water conditions for culturing should be maintained at optimum conditions for spawning. The alkalinity of Lake Superior water used in Duluth is about 42 milligrams per liter, and the hardness is about 45 milligrams per liter. In general, fathead minnows are adapted to waters of higher hardness than Lake Superior. DO levels should be maintained above 5.0 milligrams per liter by supplying continuous general aeration to the tank. It's a good policy to keep pH levels as close to natural water pH levels as possible. Maintain the water temperature at 25 plus or minus 2 degrees centigrade and regulate the photoperiod cycle for 16 hours light and 8 hours dark. Light bulbs that mimic the wavelength of sunlight stimulate the growth of periphyton, which in turn can provide fish with an extra dietary supplement. A flow-through water delivery system is the most desirable arrangement for the fathead minnow culturing system. Delivery systems constructed of threaded PVC pipe are widely used. Glued PVC may be used for drains, but not for water delivery, due to possible toxicity of the PVC glue. Do not use rubber, copper, or brass, and plastic should be avoided as some plastics may cause toxicity. Glass, stainless steel, Teflon, and PVC are the preferred construction materials. Always use three-pronged electrical equipment and be sure it's well grounded. A recirculating water delivery system can be made of the same materials, but a biofiltering system has to be added. This can be constructed of any non-toxic, high-surface area material, such as pea gravel or crushed coral. The flow rate should result in at least 3 to 4 turnovers per day. For a static system, each tank must have either under the gravel or external filtration. Every 2 to 3 days, change at least 25% of the water. Use distilled water to replace water lost due to evaporation to avoid the concentration of dissolved salts. Any larvae should be placed in a container that allows water flow while preventing them from becoming captured in the filtering system. To disinfect each tank, bleach added at the rate of 3.8 milliliters per liter of water will yield 200 parts per million free chlorine in the tank. Use this as a 1-hour static bath, then drain and neutralize with sodium thiosulfate or rinse thoroughly with culture water. A chloride test kit can be used to ensure that all chlorine is neutralized. To initiate the cultures, begin with embryos, as they're less likely to be diseased. Embryos should be obtained from reliable commercial suppliers or laboratories with existing cultures. After allowing the embryos to acclimate, incubate them with gentle aeration at 25 degrees centigrade. Larvae will hatch in 4 to 5 days after deposition. To move the fry, several larvae can be pipetted at one time with a large bore volumetric pipette. For the rearing tanks, recommended stocking density is 200 to 250 larvae per 8 to 10 gallons of water until the larvae are 30 days old. Within 24 hours of hatching, feed the larvae live brine shrimp or artemia that are less than 24 hours old. It's important to have artemia small enough for the fathead minnow larvae to ingest. The fish are fed 2 to 3 times a day, 5 days a week, for at least the first 2 weeks, and once a day on weekends. Try not to overfeed, as DO levels will drop as the shrimp decay, and remove excess food daily. Fungus growing on the waste food can cause problems for the larvae, as they can get caught in the growth. Hatching the brine shrimp is simple, and most artemia suppliers provide the conditions with the cysts. Using a round or conical flask, keep the artemia in suspension in 25 to 30 parts per thousand seawater. The artemia will hatch in 24 hours at 25 to 28 degrees centigrade. At 30 days, any fish that weren't used for testing can be weaned over to frozen brine shrimp and grown out for broodstock. 300 to 400 fish can be grown out in a 50 to 70 gallon tank. At 3 to 4 months of age, thin the population to 30 or 35 fish per 10-gallon tank. To hasten the maturation process, spawning tiles can be added to the tank. Fathead minnows begin to show signs of maturity at approximately 3 to 4 months of age. Mature males are identified by black coloration and an enlarged head with rows of tubercles across the snout. Females are smaller, olive-colored, and exhibit an ovipositor when mature. The more prolific spawners are usually between 5 months and 1 year old. 2 methods can be used for separating the fish for spawning. Brood pairs can be separated by dividing a 10 or 15-gallon aquarium into 4 quarters with stainless steel screens. A single water inlet, drain, and air stone can service each tank. Paired spawning reduces competition and allows for monitoring the fecundity of individual fish. The advantage of paired spawning is that daily records can be reviewed to identify sterile or spawned-out fish, which can be replaced to maintain egg production. A second option is to place mature adults in tanks with a female-to-male ratio of 8 to 3. In the wild, males usually select spawning locations underneath objects, and a PVC pipe cut in half or a clay tile provides a good substrate for culturing. Roughening the inner surface helps the eggs adhere to the substrate. The male will use his tubercles to clean an area on the underside for spawning. Fertilization of the embryos is external, and the buoyant embryos stick to one another underneath the substrate surface. Females release an average of 100 to 200 eggs per spawn, while larger females can release 200 to 400 eggs. They commonly spawn every 4 to 5 days, but can spawn as often as every 2 days. Reproduction should be monitored and recorded. If no embryos are recorded in a 3-week time period, replace the brood pair. Longer periods of time can be allotted for young fish that are just beginning to spawn. Feed the adults twice daily, Monday through Friday, and once on both Saturday and Sunday. The most easily used and consistent food source is frozen brine shrimp that has been partially thawed before feeding. Natural growths of algae on the aquarium may be quite helpful to the fish's diet. Trout chow or flake food can also be used as a supplement. After the fish are sexually mature, the substrate should be checked daily for embryos. To collect the embryos, check the spawning substrates in the mid-morning. This allows for early morning spawning to occur uninterrupted. To retrieve each tile, use a set of tongs dipped in boiling water between tanks to minimize possible disease transfer. The embryos can either be tested immediately or transferred to the hatching system. To hatch embryos directly from the tile, place each tile on its side in a pan that's preferably white for ease of seeing the hatched larvae. Cover the tiles with water and aerate to circulate the water. For hatching embryos in a separatory funnel, remove the embryos from the tiles with a gentle, circular rubbing action and pipette them to a separatory funnel that's aerated with filtered air. After 2 days, these embryos are placed in an aerated pan containing culture water where they'll hatch in another 2 to 3 days. The aeration of the tiles, or separatory funnels, serves to keep fungal spores from settling on the embryos. Check the embryos for fungus or lack of viability. It's critical to remove the non-viable embryos using tweezers at 24 and 48 hours. On days 3, 4, and 5, embryos on tiles should be checked, but disturbances should be minimized to prevent early hatching. If more than 50% of the embryos on a tile die, discard that tile. At a temperature of 25 degrees centigrade, the embryos should hatch within 4 to 5 days. After each use, tiles can be disinfected in a chlorine bath and the chlorine neutralized with sodium thiosulfate. The best disease prevention is to keep the tanks reasonably clean and to remove any dead, moribund, or deformed fish immediately. Abnormal behavior may indicate potential disease problems. It's critical to keep records each day to assist in the monitoring the health and fecundity of the cultures. More records may be needed for different quality assurance plans. Monitor the age of spawners and discard those over a 1.5 years old. Once the embryos hatch, transfer the larvae to a rearing tank at a density of around 200 to 250 per 8 to 10 gallon tank. One aspect of testing that's overlooked is the importance of handling the animals correctly. The larvae must be handled with a large bore pipette or eyedropper to prevent damage. Commonly, a 50 milliliter pipette with a wide bore is used. Feed the larvae several drops of concentrated Artemia naupliae that are less than 24 hours old. Be sure to adjust the feeding rate for the number of fish and feed the fish twice daily, Monday through Friday, at least 6 hours apart, to ensure that live artemia are present for the fish. Feeding only once per day on weekends has been successfully done in Duluth. As the fish grow, increase the quantity of artemia so that only a small amount remains after the fish have fed. Observation of the amount of food left is critical in adjusting the feeding rate. Larvae should also be reared as future broodstock. To ensure a good gene pool mix, larvae for future broodstock should be the progeny of as many spawning adults as possible. New genes should also be introduced to the spawning pool every few generations. This could be accomplished by crossbreeding the fish from the wild or from another laboratory. Fathead minnows are hardy and will survive under a wide variety of conditions. However, the successful culturing of fathead minnows requires a clear understanding of the biology of the fish and close attention to detail. For more information on culturing fathead minnows, consult these documents: Short-term methods for estimating the chronic toxicity of effluents and receiving waters to freshwater organisms, Guidelines for the culture of fathead minnows for use in toxicity tests, and Methods for measuring the acute toxicity of effluents to freshwater and marine organisms.